This lecture is entitled Biomedical Informatics and Covid-19. The objectives are to define biomedical informatics, to describe COVID-19, to understand aspects of COVID-19 disease surveillance, and to recognize how biomedical informatics has contributed to developing COVID-19 treatment and prevention strategies. Let's start by defining biomedical informatics, and that is an umbrella term that encompasses both bioinformatics and public health informatics, two topics we introduced in other lectures. First, computers are used in some capacity for the analysis or modeling of data, as well as to capture and display data. And second, the type of data we are talking about is health-related data of some sort, including genetics data, clinical data, epidemiologic data, and environmental data. Computers are tools for the use of mathematical, statistical, and computational algorithms to gain useful insights and organize and visualize data. Considering these issues, the definition of biomedical informatics that we have settled on for this project is that it is an interdisciplinary field that combines aspects of computer science, mathematics, and statistics to collect, store, manage, analyze, and interpret biological and clinical data to improve human health. Let's start our discussion about COVID-19 by thinking more broadly about infectious diseases. New infectious diseases have arisen throughout human history. These diseases can be caused by viruses, bacteria, and other organisms that live in our bodies. Some of them are beneficial, some are neutral, and others can be harmful. Organisms can change and evolve, and that can lead to some of them going from neutral or beneficial to harmful. Occasionally, humans encounter a new organism that causes a pandemic, which is characterized by widespread disease and death. For example, smallpox was responsible for many plagues that affected Europe even as far back as 165 AD, and it was a notorious illness brought to the Americas by European colonizers in 1492 and 1520, which resulted in the death of many Native Americans. You may have heard about the plague or Black Death that happened in the Middle Ages that wiped out a substantial proportion of the population at that time in the regions it affected. More recently, in 1918, there was a strain of the flu that infected many people and also resulted in many deaths. History has taught us that these infectious diseases can start anywhere and any time. While they are rare events, people around the world are always on the lookout for signs of them arising via government agencies and within health institutions. We know that even though we don't have to worry about them most of the time, when they do arise, they can mean serious business and a quick change in our lives. So what happens when a new infectious disease arises? We usually can't tell there is a new disease if only one person gets it. We start to notice it when there is a pattern of patients with similar symptoms in one geographic area. Or, now that there is world travel, of people who have been in contact with one another. Several things happen once we notice such a pattern. Scientists identify the organism responsible and how it spreads to hosts. Epidemiologists investigate the spread across populations. Governments issue guidelines to mitigate disease. Healthcare workers do their best to treat patients. Scientists develop new vaccines and treatments. As you can see, many people in many fields help, and biomedical informatics approaches also contribute in many ways. There are many organisms that can cause infectious diseases, but let's focus on coronaviruses, which are the type of virus that gives COVID-19. Coronaviruses are a group of viruses that can live in animal hosts. 7 of them are known to cause disease in humans. This is a figure representing what they look like under a microscope. They have protrusions, depicted here in red, on the outside that are the spike proteins that help them enter host cells. And inside a circular body, or a spherical body, formed by a lipid membrane, there is single-stranded RNA, which contains the instructions for the virus to be able to replicate. 4 of the 7 coronaviruses that affect people are known to cause the common cold. 3 of them cause severe disease and have been linked to outbreaks in recent history. SARS-CoV is one of these viruses, and it was identified in 2003 as causing severe acute respiratory syndrome. MERS-CoV was identified in 2012 as causing Middle Eastern respiratory syndrome. And as you probably know, SARS-CoV-2 is the one responsible for COVID-19, but we'll get into that next. As I mentioned, people all over the world know that they should be on the lookout for new infectious diseases arising because even though these are rare events, a serious new disease has the potential to spread quickly and be devastating to humans. A new disease could arise anywhere, so this is a global problem that requires cooperation of countries to help identify and solve problems that affect us all. In December 2019, one of these events happened where many patients had severe respiratory symptoms that required hospitalization and a new disease was suspected. A coronavirus was identified as causing the symptoms and named Severe Acute Respiratory Syndrome Coronavirus 2, or SARS-CoV-2 for short. The illness that resulted from the virus was named Coronavirus Disease 2019, or COVID-19 for short. The illness spread very quickly. We don't know who the first person or persons infected were, but we suspect it happened around November 2019. The outbreak was detected in December 2019 once there were enough people having new symptoms that we recognized something was going on. By March 2020, the World Health Organization declared COVID-19 to be a pandemic. By then, there were 118,000 cases in 114 countries and 4,291 deaths. It was clear that COVID-19 was going to affect a lot more people, even with serious attempts to control its spread. An interesting and perhaps unexpected aspect about COVID-19 is how much symptoms vary from person to person who are infected with the virus. Some people have the virus in their body but have no symptoms at all, which is one of the reasons why the virus has been able to spread so efficiently. Many people with COVID-19 have mild to moderate symptoms, which are similar to those of having a flu-like illness, including fever, cough, pneumonia, diarrhea, muscle pain, or tiredness. Some people have severe disease requiring hospitalization and admission to an intensive care unit. More severe things that can happen are kidney and/or lung failure. In the most severe situations, people can die of COVID-19. We know this from news reports, but also because most of us have had family members or friends who have passed away over the past year or so. When infectious diseases arise, one of the important things we need to figure out is how they get from one person to another. That is, how they are transmitted. Because SARS-CoV-2 was identified as a respiratory virus, its transmission route was likely that of other respiratory viruses. It might spread through contact by touching an affected person or object, through droplets, which means by being close to the exhaled breath of another person when a relatively large liquid droplet enters your body as you breathe it in, or potentially through your eyes. Its transmission might be airborne, which means it can be inside small droplets that people breathe out, and these can travel farther and linger in the air more than the larger droplets. In the beginning, the safest thing we might do is assume that it can be transmitted in all of these ways and take as many precautions as possible. Epidemiologic studies have found that most SARS-CoV-2 transmission is through respiratory droplets, although some airborne transmission can also occur, and there is a contact transmission possible as well. Once we know how SARS-CoV-2 is transmitted, we can figure out how to protect ourselves from getting it into our bodies. All citizens can take action to limit spread by following guidance on non-pharmaceutical interventions. That is, through actions that don't involve drugs or vaccines. Drugs and vaccines play an important role too, but especially in the early days of the pandemic, we weren't sure about best drugs for COVID and there were no vaccines. The non-pharmaceutical interventions slow the spread of SARS-CoV-2, which gives us some time to develop vaccines and treatments. Specifically, here are some of the things that we can all do that we learned about based on our earlier studies. Social distancing or staying far apart from one another. Wearing masks to avoid droplets being transmitted. Washing our hands often to limit the possibility of contacting the virus on a surface and then bringing it close to where it can enter our body. Increasing ventilation in any indoor space. And making sure we quarantine if we are sick or we test positive to make sure we don't transmit the virus to other people. Now let's talk about how gathering and analyzing data has been helpful during the pandemic, and let's focus specifically on some biomedical informatics contributions. How was SARS-CoV-2 identified? We did so by sequencing its genome from patient samples. As we talked about in bioinformatics lectures, DNA sequencing is when we take a sample of DNA and find out the bases that compose it, which in turn can be used to find out what genes are present in a cell. In the case of SARS-CoV-2, because it is a coronavirus, we sequenced samples to find out what its single-stranded RNA genome was. bioinformatics researchers help to analyze the data from the sequencer to identify the SARS-CoV-2 genes. Via imaging studies, we can also see what the virus looks like. Bioinformatics researchers can help reconstruct what the proteins look like in 3 dimensions based on their amino acid sequence, as well as help determine what the function of parts of each protein might be. Knowing the genome and protein structure of SARS-CoV-2 is important to create laboratory tests to determine if people have the virus. Along with sequencing the SARS-CoV-2 genome, we can compare it to other coronaviruses. By doing so, we can help identify its origin. The phylogenetic tree here is an example from a paper that used an algorithm to see how similar several coronaviruses were to each other, and based on this comparison, we can establish our best guess for their shared lineages. You can see that SARS-CoV-2 here is nearest to a few strains of bat coronaviruses, while the SARS-CoV-1 gene is nearest to some strains of civet coronaviruses. We don't know with certainty where the SARS-CoV-2 virus arose from, but based on other known strains, this tree suggests that it may have been a strain from bats that eventually made its way to a person or humans. Comparison of protein sequences across coronaviruses also provides clues about how SARS-CoV-2 causes disease similarly or differently than other strains. Here is a figure from the same study as I just showed you, the one that compared the overall genomes, but in this figure, amino acids of key protein regions have been aligned, and there is specific depiction of which ones are responsible for the binding of spike proteins to host receptors in the case of MERS-CoV, SARS-CoV, and SARS-CoV-2. These are another example of how bioinformatics researchers can help analyze data that is important to understand the infectivity of SARS-CoV-2. Another contribution of bioinformatics is monitoring how SARS-CoV-2 changes over time. By changes, I am referring to the occurrence of changes in its RNA genome. Remember from other bioinformatics lectures that all organisms can have changes to their DNA, or RNA in this case, as cells replicate. When variants switch to new ones, sometimes we term them mutations. We are interested in tracking these variants because it helps us find out how the virus is spreading. It can be that in some parts of the world a specific variant is present, and knowing where that variant was first identified can tell us about how SARS-CoV-2 is moving around the world. Several of the sequence changes don't seem to affect whether the virus gives more or less disease, or whether it spreads more easily, but some changes may actually do just that. In other words, we can also track changes over time in case a new variant arises that makes the virus more deadly or helps it spread more easily from person to person. This figure on the right is from a study that shows aligned SARS-CoV-2s from around the world that have different variants. As you can see by some of the names that are referencing different geographic areas around the world. Let's shift a bit from biology of the virus to population-level disease monitoring. In terms of biomedical informatics, we're moving from bioinformatics to public health informatics. Health departments and other agencies around the world started to track who was likely to have COVID-19 based on symptoms as soon as the illness was recognized. As tests became available that were specific to SARS-CoV-2, we were able to track based on tests, namely who was testing positive. As mentioned previously, some people who have the virus don't have any symptoms, and because of that, it is important to perform tests in many people, not just those who are sick. Another thing we do when there's an infectious disease is track those who were in contact with someone who tested positive because they are at risk for having been infected. By gathering data and analyzing it, we are also able to study the characteristics of all the people who do and don't have COVID-19 so that we can identify risk factors. That is, we can find out who is more likely to test positive, among those who test positive, who is more likely to have severe disease, and so on. Biomedical informatics experts help collect data and create applications to analyze and interpret the data, which is of interest to researchers, health providers, government agencies, and the general public. Here's an example of a website that monitors where and how many COVID-19 cases there are across the US, as well as the world. In addition to providing a map that shows where in the world there are more cases at any given time, you can also just see a ranking of where there are the most or least number of cases as well as deaths. You can go and explore this website to see what other kinds of information it provides. Besides looking at where the cases are and how many there are, we want to monitor how this changes in time. During the beginning of the pandemic, Monitoring the rate of change is what helped us figure out that SARS-CoV-2 was spreading very quickly and that things were going to get worse and action should be taken immediately. Here are some specific plots from the New York Times that show how many cases there have been since the beginning of the pandemic to March 2021 for the US and the world. Changes to plots like these over time help guide us to determine whether we should have more or less restrictions to avoid viral spread, and they are also helpful to know how healthcare systems need to be prepared for more or less patients arriving in need of care. One of the first things we noticed by tracking who has COVID-19 in the US is that some racial ethnic groups were at increased risk of COVID-19, as well as having severe disease and dying. Here's a table by the CDC that shows figures of the difference across some racial ethnic groups compared to non-Hispanic white persons. For example, persons who are non-Hispanic, American Indian, or Alaska Native are 1.9 times more likely to be a case, 3.7 times more likely to be hospitalized, and 2.4 times more likely to die of COVID-19 than non-Hispanic white persons. Identifying these disparities can help us come up with interventions to reduce them. In other parts of the world, other disparities may be observed. The numbers of cases around the world are provided by health agencies, and one of the sources of these data are healthcare systems. In many healthcare systems, patient records are stored as electronic health records, and looking at these in more detail can help us understand disease. As they care for COVID-19 patients, some healthcare providers, including doctors and nurses, might notice patterns among the patients. These patterns may lead to hypotheses about aspects of COVID-19. Analyzing large data sets, such as aggregated electronic health records across many U.S. health systems, provides us with a sample size of patients that can help us clarify whether the patterns some providers have observed are truly associated with illness. For example, we now know that lack of smell is a symptom of COVID-19, not something we had expected. We still don't know exactly why some patients with COVID lose their sense of smell, but studying data on a lot of patients, as biomedical informaticists do, can help us recognize this unexpected pattern. Further studies can look into why it's happening. Another example is trying to learn from images of lungs of people with COVID-19. Here are some CT scans that have taken images through people's chests. In the middle is the heart, and the overall dark structures are the lungs. The chest is at the top, and the back and spinal column are at the bottom. Doctors called radiologists started to notice that portions of the lungs where the red arrows are pointing have characteristics of COVID-19 infection. Biomedical informatics experts can help design algorithms to identify these regions with computer programs and relate them to COVID-19 outcomes, which may help us predict who will have worse disease and why. To summarize, expertise in biomedical informatics has contributed to understanding COVID-19 in various ways. Sequencing the genome of SARS-CoV-2 and noting mutations as the virus is transmitted from person to person, monitoring the spread of COVID-19 around the world, and identifying clinical features for COVID-19.